the materials that carry the genetic information in all living things, including viruses. Yeah. Okay. Maybe you want to recap on what is the difference between DNA and RNA? Siapa ingat lagi? What is the difference between these two nucleic acids? In term of the nucleotides, siapa yang ingat? DNA ni ada apa? Apa dia punya nucleotides? Saya nak tanya siapa ni Jom kita tanya Azhar. Nur Azhar ada? Nur Azhar Muhammad Idris. Apa dia Azhar? Do you still remember the nucleotides that present in DNA?
Is that yang adenine, guanine?
Betul.
Untuk DNA ada 4, ATCG. Untuk RNA ada 4 juga, AUCG.
Okay. Ingat tak apa tu A tu apa? Adenine, thymine,
guanine, cytosine.
Okay, betul. Dan untuk RNA kita ada uracil dia gantikan satu nukleotid daripada DNA. Okay, selain daripada tu dia punya gula pun tak sama kan. But in term of in this molecular based detection approach, we are actually play around with the nucleotide sequence. But of course, we could also play around with the other structures such as the phosphate backbone, and the nitrogen, as well as hydroxyl group in the in these two nucleic acids. Okay, so let's go this part. Okay, so basically this molecular based approach has been applied in almost all fields. As you can see, I have actually summarized in these slides. We have applied the molecular based intervention in medical field, microbiology field, biomedical science, biotechnology, food analysis, environmental analysis, and forensic science. Maybe later we can actually list down what are the applications that can be implemented in all these field, so that you can have a clear picture. Because in health science, we always focus on something that is related to disease, what else, that dealing with the treatment of the disease, but we don't know much about other application in other field of study, such as food analysis kan. Most of you do not have the exposure on this kind of analysis. What they actually do when they perform molecular based intervention in food industry, for example. So later I think we can do some discussion also on this if we have time. Okay. So listed here in this slide are a few examples of the application of molecular based approach in biomedical science and medical field. But this is not extensive. This is just a few examples of the application. For example, detection of cancer genes, diagnosis of certain diseases, detection of specific pathogens, detection of drug-resistant genes, as well as the measurement of the expression level of certain genes. So I will explain later in more detail when we go to each technique. Okay, sebelum saya sampai kat sini tadi, saya sebenarnya dah minta you all jawab kan dekat Padlet. Jom kita tengok jawapan di Padlet dulu. Okay. PCR je saya baru jumpa ni. Share any molecular based testing that you know other than PCR. Siapa tahu? Saya kena cari lagi ni, siapa lagi yang saya tak panggil ni? Oh, ada dua Nadia. Tadi saya panggil Sharifah Nadia kan? Ni Nur Nadia? Nur Nadia Muhammad Fatihi. Apa lagi agak-agak apa ni? Molecular based approach yang awak dah pernah dengar? Nadia? Or anybody?
RTK.
Tak dengar lah, Liana
RTK dah tu.
RTK. RTK tu dia sebenarnya itu ke yang saliva tu? Saliva pula. COVID test tu That one is actually bukan molecular based, dia sebenarnya antibody antigen based detection. So that is based on apa ni kata antibody antigen ni lebih kepada immunology punya ni lah field. Kita tengok antibody and antigen level, the presence of antigen inside your body. Okay. Jawapan lain saya dapat sini, nucleic acid amplification test, in vitro and in vivo. Okay, lagi ada sesiapa nak cuba lagi? Sebelum ni you all pernah belajar tak tentang PCR? Kan, since ada yang dah jawab PCR tu kan? Have you learned about PCR in any of your courses before in year 1 and year 2? Pernah tak Hayati? Dah pernah belajar tak Hayati? Ya, dalam subjek apa tu? Dr. Arifin bagi lecture. Dalam kos apa ya? Tak ingat Hayati. Kos apa? Saya nak tahu je sebab apa tu nak apa kata nak membayangkan your knowledge in what level for apa ni, for molecular punya teknik ni. Immunology. Okay. Ada lagi tak jawapan lain? Tak ada ya, nampaknya itu je. Okay, so yes, PCR is one of the molecular based technique that is currently being used extensively. As we can see from COVID test tu kan. COVID test we actually use RT-PCR. There are many types of PCR. PCR tu kita boleh apa orang kata bahagi lagi. We can divide it into many other PCR methods. This I will cover later, but just want to give you the confirmation. Yes, that's right. PCR is one of the molecular based testing, and it is actually dealing with the amplification of the gene of interest. So same like what has been answered by your friend, nucleic acid amplification ni sebenarnya related to PCR jugalah. In vitro and in vivo, okay. When we say about in vitro, it is about outside the human cell lah kan. So yes, that's right. It is in vitro. Most of the molecular based approach actually in vitro tests. And when we talk about in vivo, it is in the cell. This actually orang kata It is not so many, but yes, we have. And the example that I will give you in this lecture is about FISH, which is the abbreviation of fluorescence. Apa tu? Saya pun lupa. Nanti saya check balik. Itulah saya pun dah tak ingat kan. So this FISH technique is actually apa kita panggil use the concept of in vivo, in situ. In situ. Kita panggil in situ. In situ tu maksudnya di dalam sel tu, di dalam kromosom tu, in which the DNA will not be extracted out from the cell, from the chromosome. Dia elok dekat situ je. Okay, so this slide shows you the summary of the molecular techniques or molecular approaches that I will present in this lecture. So there are actually many other molecular-based intervention that we have out there, but today I will emphasize on these selected approaches. So as we can see here, there are southern blot, northern blot, microarray, FISH, multiplex PCR, real-time PCR, Sanger sequencing, RFLP, and NGS. Have you heard about all these terms before? Pernah tak dengar southern blot? Siapa saya nak tanya sorry, saya tak tunjuk. Okay, sorry. Share this thing. Because usually I share my window, but today I have a few videos that I want you all to watch in the YouTube. So that's why I'm sharing my tab. That's why I always forgot to switch between the tab. So I'm very sorry about that. Ni punya apa kata, saya ni noob sikit ni tentang apa tu orang kata ni, masih lagi belajar untuk lebih cekap menggunakan online punya tools ni. Okay, so alright, I have actually asked this in the Padlet. What do you know about the term nucleic acid hybridization? Okay, so when we talk about hybridization, you need to remember ligation or annealing process. Dia melekat, melekat. So we know that DNA is a double-stranded apa orang kata, rantaian. The chain is in a form of double-stranded, right? So this double-stranded fragment are actually complement to each other. So usually in vitro or in outside the cell, we are trying to mimic what actually happened inside the cell. In our body, the process we call it as DNA replication and transcription. This is happen in our body, but when we perform it outside our body, we try to mimic the same reaction like what actually happened in our body. So we call it as hybridization. Maksudnya kita nak lekatkan dia ni daripada single stranded, we anneal these two fragments together. So this is actually called as hybridization. So all these molecular based technique actually use hybridization concept. So faham ya? Sekarang ni kita dah tahu apa maksud hybridization ni. So let's have a look at the next slide, and I want to emphasize also, we know that RNA is in a form of single-stranded, but we need to convert the RNA to cDNA in order for it to become double-stranded. So because of this, we need the help of reverse transcriptase enzyme for the RNA to be converted to cDNA. Okay, so far jelas tak? Ataupun dah rasa macam susah nak keep up? Actually, I am trying my best to explain to you in a layman term so that you can grasp all these concept very easily, because all these principles are actually in a real life is very complicated, and it is difficult actually for you to understand the principle and the workflow of the process if you never conduct the experiment before. So I think my approach is that I want to explain to you in a very simple way. So I hope that you understand. Kalau ada apa-apa yang tak faham, just tanya terus. Okay. So I will start with the first technique, which is southern blot hybridization. Okay, what is southern blot hybridization? Okay, so this is actually quite wordy. I hope that you can read it first to get the idea, and then I will show you the flow of the procedure. Saya bagi masa 1 minit lah untuk baca. Baca dulu. Mungkin nak cari seorang lah untuk baca kuat-kuat ni. Shasha Shafika, can you read the what has been written in the slide? Shasha Shafika.
Get it there.
Okay.
Okay. Southern blot hybridization refers to the detection of specific DNA fragments that have been separated by gel electrophoresis. After the electrophoresis, the separated DNA fragments are denaturized and transferred to a nitrocellulose or nylon membrane sheet by blotting. In the blotting, the gel is supported on a sponge in a bath of alkali solution, and buffer is sucked through the gel and the sheet by paper towels stacked on top of the nitrocellulose sheet. The buffer denatures the DNA and transfers the single-stranded fragments from the gel to the surface of the sheet, where they adhere firmly. The nitrocellulose sheet containing the bound single-stranded DNA fragments is peeled off the gel and placed in a sealed plastic bag or a box together with buffer containing labeled DNA probe specific for the target DNA sequence. The sheet is exposed to the probe under conditions favoring hybridization. After the hybridization, the sheet is removed from the bag, washed thoroughly to remove the unhybridized probes, and viewed using autoradiography or ultraviolet light depending on the labels used, radioactive or fluorescent. An adaptation of southern blotting is northern blotting, in which RNA molecules are electrophoresed through the gel instead of DNA.
Okay, thank you Sasha. Semua orang faham tak bila baca tu? Sekarang ni saya tunjuk pula dia punya flow. Okay, kita tengok dulu. Have you learned about gel electrophoresis before? Okay, so maybe we can recap on what you have learned about gel electrophoresis. Dia guna apa tu? Agarose gel ke? Acrylamide gel ke? Yang mana satu? Yang you all belajar tu? Ingat tak?
Agarose gel.
Agarose gel. Okay. So actually we can also use acrylamide gel, but most of the time we use agarose gel. Okay. The apa kata, the preference of which gel and also the percentage of the gel is depend on the size of the target gene. So usually we already know what is the expected size of our gene of interest. So in this case, for southern blotting, we use agarose gel. So at first, we need to have the DNA sample. So usually at this step, the DNA will be extracted out from the cell. Then you will perform the gel electrophoresis. At this stage, you should get the DNA fragments from several ways. It's either by using a restriction enzyme. Restriction enzyme, usually we use the abbreviation of RE. RE. So RE will cut the DNA. You just imagine that this DNA is in circular form. Dia bulat kan? Bulat. So when we add in restriction enzyme, this restriction enzyme serve as scissors. Dia macam gunting, dia potong. Dia potong. So this DNA will become many fragments. And these DNA fragments can also be derived from PCR. When you perform PCR, you will get the PCR product at the expected size that you want. So then you will perform gel electrophoresis. So gel electrophoresis is actually to separate the fragments of the DNA based on the size. So it will go from the negatively charged to the positively charged arrangement. So from at the bottom, the size is the largest, and it will go down based on the size of the fragments that you actually cut or amplified using the PCR previously. Okay, at this stage, you can see maybe there are more than one, or maybe there is only one fragment. So it depends on the results on the agarose gel. You can actually view the band. Where is the band on the gel electrophoresis when you view the gel under UV light? The gel is actually like the one that I've shown just now. Ni. Ini rupa gel tu. This is the gel, and you cannot see actually the band. After you perform the gel electrophoresis, you cannot see it with your naked eye. Kat mana band tu? Kita tak nampak. So you need to have the assistance of the UV light to observe it, because this to view the gel agarose, you put in the dye. This dye is it has a fluorescent dye in it. So in order for you to view the gel, you need to view it under ultraviolet light. But the step will not end there. Dia ada lagi banyak step. So for southern blotting is actually a process where you transfer the DNA fragment yang tadi tu kita dah susun, fragmented tadi tu, kita dah susun ikut dia punya size. On the gel electrophoresis, we want to transfer the DNA onto a nylon paper. It can be nylon filter paper. It can also be nitrocellulose filter paper. So these two filter paper are the most common blotter, yang kita panggil blotter, or the filter paper that we use to transfer the DNA from the gel onto the paper. Kita nak transfer dia macam dia akan jadi mirror image. This DNA band will be transferred onto the nylon paper, and then it will become mirror image of the initial gel tadi tu. So far, are you understand? Boleh faham Okay. So this is the process lah tadi yang kita panggil blotting tu. Kita letak paper towel, ada sponge. We make it like a sandwich lah. So we can actually apa kita panggil lah, transfer the DNA. Kalau bahasa Melayu tu kita panggil tekap, kita tekap dia supaya dia melekat. Okay, so now this is the replica of the DNA on a filter paper, and then later there will be an addition of radioactive probe. So this probe is tagged with the radioactive labeling lah kita panggil label with the radioactive tag. So with this tag you can visualize later using auto radiography. So when you put a radioactive tag, you need to observe the results using X-ray film. But if you use a fluorescent tag, you can visualize the result using fluorescence detector. So it depends on the tag that you use and also the availability of the imaging system that you have. Kalau kita ada yang jenis X-ray tu, kita kena guna radioactive labeling lah. But if you have an imaging machine that has a fluorescent detector, you can use fluorescent tag. So this fluorescent tag probe, for example, or radioactive probe, it will hybridize to the complement nucleotide. Kalau complement ni maksudnya dia macam ni. Kalau complement ni dia saling melengkapi kan. So if the sample has A adenine, then if the probe has T thymine, it will bind, it will hybridize. Faham If the DNA has C cytosine and the probe has guanine, it will bind. So this is what is meant by complement. So when they complement each other, they will bind and it will give these results. So kita nampak lah, initially you can see there are so many bands when you run the agarose gel electrophoresis. But later in the end, you will see only the band that is bind or hybridized with the probe. Okay, actually I want to show you what is the look of the what is the example lah of the real photo. Kejap saya search dulu. Southern blot. Southern blot results. So you will see something like this. Ada tak yang before and after? Ini tengok sekejap. Saya tak jumpa, but okay, just look at this example. So this is the real photo of the kita panggil auto radiography. So the one with this intact band shows that the DNA band a nail to the probe. Yeah. Okay. So I will continue with this slide. Okay, as mentioned earlier, in this note, northern blotting. Northern blotting is when you use RNA as your starting material. So the difference between southern and northern blot is the starting material. Southern blot kita guna DNA as the template. Northern blot we use RNA as the template. Okay, jelas tu soalan tu saya tanya di Padlet dan saya dah bagi jawapan. Tapi ada satu lagi yang saya tulis di situ, Western blot. Okay, although we could see that the name is like apa orang kata kutub kan, kutubnya utara, selatan, timur, barat kan. But the name is actually derived from the founder of the technique. Saya lupa nama depan dia tapi Southern is the surname. Jom kita tengok itulah saya pun tak ingat juga. Mungkin siapa yang dapat cari tu boleh share juga pada kawan-kawan siapa nama founder dia ni. Southern blot. Okay, Edwin Southern. Edwin Southern. Yang menjumpai teknik ni dia yang mencipta teknik ni. Okay, back then in 1975. So, who knows what is the difference between Southern blot and Western blot? Dia ada satu lagi. Siapa nak tolong jawab? Saya nak cari nama siapa ni tadi yang saya belum sebut ni. Najiah. Najiah ada?
Ada.
Okay Najiah, mungkin awak boleh teka kot. What is the difference between Southern blot and Western blot?
Starter dia.
Apa dia? Tadi kita dah tahu Southern blot ni DNA. Dia punya material dia kan. Northern blot tadi kita guna RNA. How about Western blot? Agak-agak siapa yang tahu nak teka? Syahidah?
Antibody.
Saya ada dengar tadi tu orang sebut. Okey, dia bukan antibody tapi protein. Saya ada dengar tapi saya tak sure siapa yang sebut. Sebenarnya untuk protein. So, all these three, whether it is Western blot, Northern blot or Southern blot, the procedure are all the same. The principles are the same but only the starting material is different. So, ingat tu je. Okay, kita tengok balik flowchart tadi. So dia punya step ni tadi. So the step is all the same. But of course we need to adjust kata in term of the process a bit. If the starting material is different. For example, for gel electrophoresis, if you perform northern blotting, you need to perform denaturing agarose gel electrophoresis. You need to add, if I'm not mistaken, formaldehyde to the gel when you prepare the gel, because you need to denature the RNAs before you perform the gel electrophoresis. And for protein, you need to perform it using SDS pitch. not using agarose gel. Yeah, SDSPH is another method of electrophoresis where use SDSPH is stand for sodium dodecyl sulfate polyacrylamide gel electrophoresis, where you use polyacrylamide gel to separate the protein of interest. But all other steps are the same. Yeah, punya flow dia semua sama. Okay. And I just want to add here, when you, I think after this step, when you already completed the addition of the probe, you need, before viewing, you need to wash the any impurities that actually not involved in the binding of the probe to the DNA. For example, maybe there is excessive reagents or even excessive probe. The probe do not bind to the DNA. So we need to remove all these impurities before we can view the X-ray film. Yeah. So I think that's it for southern blotting. Then we go to the second technique, which is RFLP. That is stand for restriction fragment length polymorphism. So kita tengok kat sini kan, ini dah nampak dah sebenarnya contoh macam mana rupa dia ni. Lagi banyak dia punya DNA fragments kan. So easy for you to remember for RFLP, it is actually combining the concept of southern blotting that we have learned earlier with the fragmentation using DNA. Yeah. So this time, sekejap ya, saya tengok sekejap ni. So, kenapa besar sangat? Sekejap, saya kecilkan sikit. Okay, so this is the overall step of the RFLP. So if you can see, the difference of RFLP is only here at this part. Yeah, with apa tu tadi, southern blotting. Yeah, so for RFLP, it actually exploit the variation of a polymorphism inside the living cells. For example, have you heard about the term allele before? Pernah tak dengar allele? Mungkin belajar masa genetik ke? Allele ni kita define dia sebagai diversity of variants in a given gene. For example, for ABO blood typing, we have blood type A, B, AB, and O. So this blood typing is controlled by ABO gene. And overall, there are actually 6 variants that we call as allele. So, as for RFLP, we are also dealing with apa kita panggil multiple allele that we call as polymorphism. Okay, the example of polymorphism is like the gene that encode for the color of your eyes. The allele actually multiple. It involved in more complicated kita nak panggil meiosis or mitosis reaction that occur. So by using this technique, several application can be conducted. And actually for RFLP, before this, back in 20 years ago, it is widely used to detect or to identify the genetic disease. For example, if somebody has thalassemia in the family, so they want to know if the baby or the kids that they have are actually inherit the thalassemia also or not. So by doing RFLP, they can have the what we call as a genetic fingerprinting patterns. So this family could locate whether that their children are actually carrying the same genetic disease or not. Based on the DNA fragment pattern that they observe from the RFLP. And other than that, RFLP is widely used in forensic science to identify the sample from crime scene. For example, in rape cases where this forensic officer, they could find the semen or maybe the blood of the victim or the apa kita panggil that culprit that did the crime. So based on these RFLP results, they could actually compare in the database. Di Malaysia, saya tak sure whether we have this kind of database or not, but in overseas, in other developed country, they are having this database, so they can actually compare straight away who is the person. And also for paternity, if the somebody want to know who is their biological son, daughter or father, RFLP is actually very widely used before. But the challenge of RFLP is that to perform this analysis, it require a longer process to complete the analysis. Maybe it will take more than months to complete, and also the procedure is quite tedious. And if you are not properly trained, it is you could cause a false results. And also it require larger sample size as compared to other techniques. So this RFLP is kind of obsolete right now because we have many other techniques that is more advanced, cheaper and less tedious as compared to RFLP. So do you want to ask anything up to this stage? Ada tak nak tanya apa-apa yang tak faham? Tak ada semua jelas? Tandanya jelas lah tu. Okay, terima kasih Nafhan. Okay, so now we go to the third techniques, which is fluorescent in situ hybridization. FISH FISH singkatan dia. Okay. So fluorescent in situ hybridization is a molecular technique for detecting and locating a specific DNA sequence on a chromosome. So it relies on exposing chromosomes to a small DNA sequence called a probe that has a fluorescent molecule attached to it. The probe sequence binds to it corresponding sequence on the chromosome. So you can imagine now that, okay, when we the term in situ is actually refer to the chromosome, whether there's no additional step that is needed to extract out the DNA. the DNA is remain at the chromosome. So usually this step is conducted on the slide, glass slide. And as the name implies, we use fluorescence. The probe is tagged with fluorescent, which apa kata, will be serve as a detector lah nanti. So we need to view using fluorescent microscope. Okay. So this technique is often used for finding specific features in DNA for use in genetic counseling, medicine, and species identification. Okay. Maybe you don't understand when I put there genetic counseling. Fish can also be used to diagnose disease, to what we call as to give the prognosis of the disease, meaning that it could actually provide knowledge on what actually will happen next to this particular person if she or he possess this disease. And also it could also give you the details on whether there is remission meaning that it is there is a reduction in terms of for example, if that person has cancer, whether that cancer is reduced, ada apa ni, improvement ke tak in term of the enlargement of the cancer cell. So this can be done by using fish. So if let's say the person has cancer, so fish could be used as a way for doctors to provide the counseling to the family members and even the patient on what to expect in terms of psychological effect on their emotion. So usually this can be done when they show the result of fish. Sebab fish ni kita sebenarnya visualize. We do not give quantitative data, but we just give qualitative data. So I will show to you what actually the look of these results for fish. Okay. Fish can also be used to detect and localize specific RNA target in cells, circulating tumor cells, and tissue samples. Okay, so for RNA, it can be used for both DNA as well as RNA. As for RNA, it can actually show the expression of the cells. So I've put here cytogenetic technique. So fish is one of the cytogenetic techniques. And the advantage of this technique as compared to other scientific technique is that it is not dealing with the living cells, because the chromosome, the sample is actually dimatikan lah. The cell is not we cannot culture it anymore, because in other type of cytogenetic testing we need to maybe grow the cells, but in fish no, because it needs to be exposed with formaldehyde where the cell will become death lah. Dah tak hidup lagi lah. So I show you this example of how fish could identify bacterial pathogen. So basically it start with collecting the sample from patient, and then the process of synthesizing complementary oligonucleotide for suspected pathogen with fluorescent tag. So proceed with chemically treating the tissue sample to make the membranes of all cells permeable to the fluorescently tagged oligonucleotide. So at this stage dia dah mati lah sebab kita treat dengan a very strong chemical, and then proceed with tagging with fluorescent complementary oligonucleotide to the sample, and the fluorescently tagged oligonucleotide will bind only to the pathogenic DNA because this probe is very specific, just like other molecular technique where it will complement to only the pair, dengan pasangan dia je yang dia akan hybridize. Macam saya mention tadi, adenine to thymine, guanine to cytosine. Okay, then we will observe using fluorescent microscope. Okay, ini dah method yang keempat. So saya nak tunjuk macam mana rupa dia. Sekejap kita buka fluorescence. Okay, check image. So macam ni lah nanti kita akan nampak rupa dia macam ni. So mana yang ada probe tu attached to that particular gene, it will fluoresce. Okay. So now I will move to microarray. So microarray is also known as a gene chip or biochip. Microarray is a hybridization of a nucleic acid to a very large set of oligonucleotide probes, which are attached to a solid support such as glass, plastic or silicon chip, forming an array. So it has two major applications, which are in gene expression analysis as well as in genetic variation analysis. And this technique is also fast and cost effective because it can analyze a very high quantity of sample in a very small apa ni kita panggil glass slide tu. So nanti kita tengok macam mana rupa glass slide tu. So the challenge of this method is that the optimization of the hybridization condition for many probes may be challenging. Moreover, the designing of probes and arrays is very time consuming. So the difficulty is in designing the probe. So we need to design a many probe. Kalau tengok dekat sini kan nampak ya, dekat atas glass slide ni dia ada banyak dot-dot kecil yang penuh di atas glass slide ni. So in each well you need to design different probe for that. So how you need to just imagine how many probe that researchers need to design in order to do this. But if they can get what they want in their experiment, then they can have like a many data for them to present. They have so many results. Okay, I don't want to show you this. You can just read this slide later. But I want you to watch the video so that you can actually have a view on how this microarray is all about. Yeah.
This animation will demonstrate how DNA microarray experiments are performed. DNA microarrays, sometimes called DNA chips, reveal the expression of thousands of genes on a solid surface, such as a microscope slide. In this example, we'll use yeast as a model system to illustrate one use of microarrays. One common use of microarrays is to determine which genes are activated and which are repressed when two populations of cells are compared. Every gene is measured simultaneously. As an example, we'll compare what happens to yeast genes when cells are grown in aerobic versus anaerobic conditions. The cells grow and adjust which genes need to be activated or repressed in order to survive. Now it is time to isolate the mRNA from both populations of cells. The cells are spun in a centrifuge. Now that the cells have gathered in pellets, we remove the liquid, but not the cells. Next, it is time to extract the mRNA from the cells. When we add the extraction buffer, the mRNA is released into the solution. Next, we remove the RNA and place it in a fresh tube. Now, let's make the cDNA from the mRNA. Here we see 3 of the many mRNA molecules from each tube of cells. Each mRNA is converted into red or green colored cDNA. When the colored cDNA is made, the mRNA degrades. Then we combine the red and green cDNA, mixing both colors into a single tube. At last, it's time to look at the DNA microarray. In our experiment, a microarray or DNA chip contains about 6,000 spots. Each spot is a different yeast coding sequence from a different gene. Let's choose 3 spots at random to follow in detail. Each spot is made of DNA that can base pair with its complementary cDNA. Here are partial sequences from each of the 3 spots we are observing. Now, let's incubate the mixed cDNA with the DNA chip. For the sake of our example, we'll zoom in and show that some of the labeled cDNA have bound to the DNA in the spots and formed base pairings. Here we see green and red cDNA bound to this spot. Only red cDNA is bound to this spot. And only green cDNA bound to this other spot. In a real experiment, you would not see any of this detail. You would only see the original microarray. Now we must wash off the unbound cDNA to see what is bound to the microarray. Let's detect the bound cDNA so it can be visualized. We begin by placing the microscope slide containing the microarray inside a scanner. We'll examine the next phase of the process, keeping our focus on the 3 spots we've been following. First, a green laser scans the microarray. The resulting image is stored on a computer for later analysis. Now it's time for the red laser. This image is also stored on a computer for later analysis. Now we move to the analysis phase. After we eject and safely store the microscope slide, we retrieve the red and green images from the computer and create a merged visualization. In the merged image, we see an aerobic gene labeled in green, an anaerobic gene labeled in red, and a gene labeled in yellow that was expressed in both aerobic and anaerobic conditions. This is one example of how DNA microarrays are used. In an actual experiment, quantitative analysis would be conducted on all 6,000 genes.
Okay, so I hope that the video that we watched just now help you to understand more on the process that actually happen in the microarray. So kita nampak dot-dot macam ni kan, the color will indicate whether the expression is actually happen or not. So far, do you want to ask me anything about microarray? Semua faham? Nanti boleh tengok lagi video lain. Saya rasa ada banyak sangat video yang membantu, because when you just focus on what has been presented in the lecture, I think it is not enough to enlighten you. So you need to have more knowledge. So it is your own effort to get the knowledge, apa orang kata, based on your needs. Because sometimes we need to observe the video for you to be more understand on the topic, and sometimes you need to do the practical in order for you to understand more. Setiap orang tu tak sama kan, but there is actually a person who can grasp many input by just listening to the lecture. Itu pun dah memadai. So it's up to you. So to what extent you want to know more about this, and I hope that you put more effort in getting the knowledge by using other resources that you can find out there. Okay. Alright, so the detection, I just want to mention here because I think that it is not covered in the video just now. Okay, so there are many type of detection that we can use for this microarray technique. We can use a fluorophore or silver or chemiluminescent label target to determine the apa orang kata, the number lah, the number of the nucleic acid that actually present in the target. So fluorophore ni basically kita guna ni lah fluorescent. Fluorescent. Silver is another technique where we use there is a chemical, if I'm not mistaken, silver nitrate. Usually it will cause the appearance of like a brownish, orange or merah-merah sikit lah, the red color on the detector. While chemiluminescent, it involves the reaction with a chemical that luminous. Dia macam apa yang kita panggil tu, macam the glow stick that actually glow in the dark. That one is using the concept of chemiluminescent. Okay, so now we move to another molecular based technique, which is gene sequencing. Siapa pernah dengar pasal gene sequencing? Saya nak cari siapa ni yang saya tak tanya lagi. Saya risau you all mengantuk je ni, tu yang saya tanya soalan tu. Nadia Nur Shifa. Berapa orang Nadia ni? Macam 3 orang pula saya dah jumpa. Ya. Okay. So Nadia Nur Shifa, pernah tak awak dengar pasal sequencing?
Nadia pergi tandas, doktor.
Okay.
Nadia
Siapa
pergi
yang jawab tu? Alia. Okay, tak apalah. Dia pergi tandas. Siapa nak tolong dia? Pernah tak dengar? Saya tanya pernah je ke? Pernah atau tak je?
Pernah, doktor.
Pernah, doktor.
Okay. So, apa yang you all faham tentang sequencing ni? Tak ada dah nak jawab.
Macam order of nucleotide dekat DNA tu.
Sebenarnya dia nak baca lah, dia nak baca sequence tu. Kan kita nak tahu apa turutan dia, jujukan lah kita panggil dalam bahasa Melayu dia, jujukan, sequencing ni jujukan. Okay, so kita nak tahu apa, kalau macam kita buat kita tuliskan contoh medical, kita tahu kan MEDICAL. So macam mana pula dalam DNA tu, apa dia punya susunan jujukan of the nucleotide sequence. So how we want to know it, we need to sequence lah. We need to sequence the DNA sample. So for RNA, we need to convert it to cDNA for us to know the sequence of the cDNA. Okay. There are actually many types of gene sequencing. Dia macam ni, yang saya nak tunjuk kat sini, Sanger sequencing and NGS. So NGS actually is more high tech lah as compared to Sanger sequencing, and NGS can be also divided into other type of sequencing strategy. Dia punya beza tu mungkin in term of dia punya apa kita panggil teknik. Contoh kalau macam kita buat macam kita masak lah, macam nak kata kalau kita masak, kita ada guna air fryer, kita ada guna kita ada goreng, kita ada guna oven, kita ada guna microwave kan. So ada banyak sangat device yang kita guna. So something like this also lah when we divide this NGS. There are many kind of apa tu mereka panggil teknik. Bila satu teknik yang berbeza je, dia namakan lain. Dia pecahkan lagi. Ya. Saya harap you all dapat imagine lah macam mana yang saya masukkan tu. And other than Sanger and NGS, we also have metagenomic sequence analysis. But this one I actually not covered in this lecture. So I just focus on these two first. Because I don't want you to confuse, because there are so many type of method. Yeah. So for Sanger sequencing, biasanya kan kalau ada nama macam ni mesti dia punya founder. Sanger sequencing, the Sanger is the name of the founder lah, Frederick Sanger, yang menjumpainya back at 1977. Kalau ikutkan tak adalah lama sangat kan. Sebenarnya tak adalah lama sangat, but the technology is progressing very rapidly. Setiap tahun ada je tambahan-tambahan lagi. The improvement that they make for this sequencing reaction. And for Sanger sequencing, we could consider it as a gold standard in sequencing technology. And it is widely used, especially in research, and in other country, it also be applied in clinical field of study. Di Malaysia, kita ada banyak juga sequencing machine yang yang ada di universiti-universiti dan di research institute. And there is also there are several companies that actually provide service for sequencing because they have the sequencer. The sequencing machine is expensive actually. Usually for us, if you want to do research that involves sequencing, we will outsource the sample. Kita hantar sample tu ke company, they will sequence the sample for us, and we will analyze the sequencing results that we've obtained from them. Sebab dia mahal ya, kita tak mampu, tak semua tempat mampulah. Okay. Sanger sequencing is suitable for sequencing single gene, meaning that the sample that we prepare for Sanger sequencing must be in a pure form. Dia kalau katalah kita ambil untuk identify bacterial species, it must be from a one bacterial colony. We cannot mix, we cannot take it, okay, contohnya lah awak ambil sampel daripada air kan, awak ambil air, lepas tu awak extract genomik terus daripada air tu, padahal awak tahu mesti ada lebih daripada satu microorganism or bacteria in that water sample. So you cannot use the sample for Sanger sequencing. You need to make sure that the sample that you've sent for Sanger sequencing is in a pure form. Dia daripada satu source sahaja. Itu untuk Sanger sequencing. So it can also sequence 1 to 100 applicant targets at the lowest cost, because as compared to other sequencing such as NGS or metagenomic, this one is the cheapest one. Paling murah lah, sebab NGS and other methods, it is meant for high throughput analysis which involve many samples at one shot. So rugilah kalau kita ada sampel yang sikit je, kita hantar untuk NGS. Tak sesuai. So if you have less than 100 or and below, you just use Sanger sequencing to perform the sequence analysis. And Sanger sequencing can sequence up to 96 samples at a time. Dan kita boleh gunakan biasanya untuk microbial identification. And it is also used in concurrent with NGS. When researchers perform NGS, usually they will validate using Sanger sequencing. So ini pun saya nak share juga video. Sekejap. Tadi you all dengar kan bunyi? Ada bunyi kan? Ada kan bunyi?
Ada.
Ya? Oh, sekejap. Saya slowkan saya punya.
Hey guys, quick back in Mr. Basics here. Let's talk about Sanger sequencing method. Now, before moving further, this video is divided into 3 parts. The first part explains the principle of the method. The second part is about classical Sanger sequencing that was used widely in 1980s. And the third part is about modern Sanger sequencing that uses fluorescence dyes. Sanger sequencing is based on dideoxynucleotides in DNA polymerizing reaction. Let's understand what are dideoxynucleotides. The nucleotide has 3 components: the nitrogenous base, which can either be A, T, G, or C, the sugar residue, and the phosphate attached with the sugar residue. When all 3 components are present, it's called nucleotide. If the phosphate is absent, then it's called nucleoside. Now watch carefully. If the hydroxyl group at the 2' end of the sugar is removed, then it's called deoxynucleotide. The deoxynucleotides are present in the DNA, and they are used as a substrate by the enzyme DNA polymerase. All the DNA polymerase, whether it's DNA polymerase 1, 2, 3, Taq polymerase, etc., uses DNTPs as their substrate. The hydroxyl group attached at the 3' end plays a very important role in DNA polymerizing reaction. During DNA polymerization, there is a reaction between 3' OH group and the phosphate of the new DNTP. In 1977, Frederick Sanger had a brilliant idea. If the 3' OH group of the nucleotide is absent, then there won't be addition of new nucleotides and the reaction would stop. The nucleotide having 3' hydroxyl group absent is called dideoxynucleotide. Di means 2 and deoxy means absence of the hydroxyl group. Let's see classical Sanger sequencing method. The template DNA to be sequenced is divided into 4 tubes. Each tube has a primer that will bind to the template DNA. All the 4 DNTPs, that is the ATP, the TTP, the GTP, and the CTP are present. Along with these, DNA polymerase enzyme is present that will add DNTPs on the template DNA. Now here comes a very interesting part. This reaction mixture also has one radiolabeled dideoxynucleotide at a very low concentration. What I mean by this is that tube 1 will have dideoxy ATP, tube 2 will have dideoxy TTP, tube 3 will have dideoxy GTP, and tube 4 will have dideoxy CTP at a very low concentration, and all of them will be radiolabeled. Now let's see what happens in tube 1. The DNA polymerase would add DNTPs on the template DNA and carry out normal polymerization reaction. If somehow the dideoxynucleotide is incorporated, then the reaction suddenly stops. The reaction terminates as there is no 3' hydroxyl group for the addition of new DNTPs. The incorporation of dideoxynucleotide and termination of polymerizing reaction is a random process. Hence, the overall mixture will have several fragments that has experienced chain termination. Now, if the length of the fragment is known, then the location of adenine in the DNA can be estimated. The length of the fragments can be obtained by performing polyacrylamide gel electrophoresis and autoradiography. Once the autoradiogram is obtained, we can now have the sequence information of the desired DNA fragment. Okay, the first nucleotide is gonna be A. The second nucleotide is A. Third is G. Fourth is C. Fifth is T. Sixth is T. Seventh is A. Eighth is G. Ninth is C. And 10th is again C. Now let's see modern Sanger sequencing method which is used even today. Now in this method, each of the dideoxynucleotide is attached with a fluorescence dye. The fluorescence dye can either be yellow, green, blue or red. Instead of having an X-ray film placed on a gel, we'll now have a gel that would give fluorescence when exposed to UV light. As each of the dideoxynucleotides have their own fluorescence, it is now possible to carry out the reaction in one single tube rather than 4 different tubes. The chain termination reaction would result if dideoxynucleotide is incorporated in the DNA. The chain-terminated fluorescence fragments can be separated by polyacrylamide gel electrophoresis in a single well. The more advanced system uses capillary electrophoresis for the separation of fluorescence DNA fragments. These capillaries are further equipped with CCD or charge-coupled device for the detection of fluorescence signals.
Okay, tak stop pula. So the video summarize on the difference between conventional lah tadi, conventional and the modern one, yeah. So basically what I could summarize in the video is that the difference between the modern and the conventional is that the conventional need to use separate tubes, right? 4 tubes, different 4 tubes, because they are not using the fluorescent dye, kan? And in modern sequencing, Sanger sequencing, I think most right now are using this modern sequencing technique lah. They are using fluorescent dye. So instead of using 4 tubes, they are merging all this tube into only one tube, because this fluorescent dye is very unique. It has different color for each nucleotides. Yeah. So based on the electrophoresis, they could identify what are the sequence of the gene of interest. As of this slide shows here, kalau macam ni kan, T lah dulu, T, C, G, A, T, yeah. And usually when we have the results, the company will give us in a form of chromatogram. Chromatogram nanti ya, saya tunjuk macam mana chromatogram, as well as the nucleotide sequence lah. Chromatogram. So macam ni nanti kita akan dapat result tu. So we will get this kind of result as well as the sequence of the nucleotide sequence. So you need to actually analyze. After you get the sequences, you need to analyze the results using bioinformatic tools. Yeah. Okay. Now we continue with NGS. So this one you can just read later. So for NGS, actually I have explained a bit when I discuss about Sanger sequencing. As I mentioned, NGS is a high throughput DNA sequencing. where it actually was introduced in 2005. Yeah? So the advantage of NGS is that it can be used for many genes, more than 100 genes at a time. And usually the application is that to get novel variants of microbial diversity. For example, novel variants of microbial diversity, so they can just use, like I've mentioned earlier, when they sample from water, uh, the DNA can be extracted straight away. And this sample of DNA possess not only one microorganism, but there are so many microorganisms. And this NGS could actually amplify the gene of interest of all these microbial communities in that water sample. But the probe or the primer that is used is very short as compared to Sanger sequencing, and because of that, Sanger sequencing is needed to actually validate the result of NGS. NGS can get so many results, but Sanger will later apa orang kata sequence the let's say saya nak bagi you all faham ni macam mana. Okey, kita dapat banyak lah beratus-ratus jenis bakteria ke contoh kan, bila kita run using 16S primer. 16S primer is actually the primer that we use to identify bacteria up to species level. For example, if you use if you want to identify apa kata up to species level macam Staphylococcus aureus, Escherichia coli, dia sampai ke belakang tu, dia bukan Streptococcus species, macam tu ya. So when this NGS got so many data, we are not sure actually whether this is accurate or not, because the primer is very short. It may be not specific. So we use Sanger sequencing to confirm the results. Yeah. So sebab Sanger sequencing, we have like a apa kata larger or longer DNA primer, yeah? Oligonucleotide primer. Okay, so and NGS is very sensitive. It can be applied to a very small concentration of sample. NGS has been applied in so many fields. Yeah? So basically, this is the step for NGS. 4 step ni adalah 4 step yang asas. Tak kisahlah jenis macam mana pun mesin tu. There are so many machine out there using different kind of algorithm. Okay, but this is the basic sequence of the flow of that NGS procedure. The first one is to construct the library. So in order to construct the library, the DNA need to be fragmentized. Yeah? When we say fragmentation, meaning that we make the DNA shorter. So how we want to make the DNA shorter? By using a physical shearing method. Physical ni whether we use equipment, equipment such as stone and mortar, or we use chemical reagent. If using digestion technique, we use restriction enzyme, or we amplify using PCR. And then after the fragmentation was done, the DNA fragment will be ligated with the adapter at each end of the DNA fragment. Dekat depan dia dengan kat belakang dia, kita tutup dia dengan adapter. And then the next step is by clonal amplification, where there is an attachment of the DNA fragment to a solid surface. In order to get the signal, a better signal. Okay, each DNA molecule in the library is bound to the surface of a pit or a flow cell and PCR amplified to create a set of identical clone. Kita amplify lagi guna PCR ya. Sebab kita nak perbanyakkan lagi kita punya template tu. Okay, so the third step is we need to perform sequence library. All the DNA in the library is sequenced at the same time using a sequencing instrument. This is a cycle with common steps. DNA-based synthesis on single-stranded DNA followed by detection of the incorporated base. Ni adalah proses di mana sequencing tu berlaku ya. Okay, and then the last step is to analyze the data. So NGS, we have like a very complex data to be analyzed. Yeah, so it depends on the type of the machine and the algorithm that they use. And because of this complexity of the NGS, usually we need to have like a skillful or trained bioinformatics specialist to perform the analysis. Okay, sebab kita dah tak cukup masa ni, saya cuba cepat sikit. I hope that you can understand my explanation. Kalau tak ada apa-apa, kalau ada apa-apa yang tak faham, just interrupt ya. Okay, I think this is the last technique that you have mentioned earlier when you learn immunology with Dr. Arifin. He had mentioned about polymerase chain reaction, which is abbreviated as PCR. So PCR is extensively used because of its rapid and easiness. It is also involve low cost equipments as compared to others lah macam sequencer NGS semua tu, itu sangat-sangatlah mahal So these are the PCR components that you need to have when you want to perform PCR. DNA sample, a set of primer, forward and reverse, nucleotides with all 4 bases, tag polymerase. Okay, tag is stand for Thermus aquaticus. Thermus aquaticus ni adalah sejenis bakteria that is a very heat resistant, meaning that this bacteria possess enzyme polymerase that can withstand at higher temperature, and because of this characteristic, this enzyme polymerase can be used for PCR reaction. Untuk PCR ni kita kena guna suhu pada 94 darjah Celsius untuk kita denaturekan, pisahkan DNA from double stranded to single stranded. So we need to ensure that the polymerase that we put in is still functioning at higher temperature, and because of that we use tag polymerase. Tag polymerase, the function is to help to synthesize a new nucleotide. Kita nak panjangkan lagi When the DNA sample is separated, dia dah dipisahkan kan, dia jadi single stranded, then it will bind with the primer. Primer tadi tu yang complement with the DNA sample will bind, will anneal, will hybridize. Semua maksudnya sama kan? Okay, dia akan melekat pada DNA sample kita tu. Primer ni pendek, so we need the help of these nucleotides tadi ni, CA, GT tadi ni, and also tag polymerase to perform the elongation. Kita nak panjangkan lagi DNA sample ni. Kita punya DNA ni supaya dia lagi panjang. Kita dapat amplifykan the gene of interest according to the set of primer that we design to that DNA tu. And then all this reaction is happen in a PCR tube containing the specific buffer. Usually we use either phosphate buffer or tris buffer. So PCR tube ni kecil aja. Biasanya the total of the reaction could be as low as 10 microlit and up to 300 microlit. Biasanya kita buat tak sampai 300 pun, mungkin dalam 50 microlit pun dah cukup untuk kita run the PCR reaction. Then this PCR tube will be put in the thermocycler. The process will be undergone in the thermocycler. So this is the process for PCR. Macam saya bagi tahu tadi, the first step is denaturing step. Ni dia tunjuk 95 kan. Biasanya kita buatlah dalam 95 ke 94 macam tu. because we want to separate the double-stranded DNA into single-stranded. Then the process of annealing where primer will bind to the template. Okay, the temperature is optimized. Tak semestinya 55. It optimized based on the results lah. Kadang-kadang kita akan kira dia punya expected melting temperature. Kita boleh tahu sebenarnya daripada the primer that we design, we can actually predict the melting temperature. So when we can predict melting temperature, we can actually predict the annealing temperature. Biasanya kalau orang yang tak tahu what is the annealing temperature, they will do the optimization using different temperature for annealing step tu nanti. So in the end, they will observe the result. Which result will come up with the best results? Yang mana yang boleh dapat DNA amplification yang paling cantik. Itu yang dia akan gunakan sebagai the optimum temperature for annealing. Okay, so the next step is extension. Ini yang saya bagi tahu tadi tu, kita nak panjangkan lagi tu. Kita nak panjangkan lagi DNA tu sampailah dia lengkap. Forward and reverse tu dia apa kita panggil? Dia bertemu lah. Dia bertemu I hope that you can imagine. Ini kalau macam ni kan, ada arrow kat sini. Ini kita bayangkan forward primer. Yang ini kita bayangkan reverse primer start dekat sini. So it will extend. Forward ni dia extend ke belakang, reverse ni dia extend ke depan. Then it will produce double-stranded DNA that complement to each other. And the process will be replicate. Ada banyak kali ni, replication. Okay, sometimes can be up to 40 cycles. So in the end, the gene of interest will be amplified in many copies, hundreds and thousands of copies after the certain cycle. Okey, inilah rupa dia sebenarnya. PCR tube tu kecil aja. Nak tunjuk kat sini kecil aja. So this is the example of the thermocycler. Ada banyak jenis lah thermocycler, and this is the look of the PCR product after you perform gel electrophoresis. Bila kita dah run gel macam ni bila kita dah run PCR, before and after dia macam ni juga rupa dia. So how you want to know whether your PCR reaction is successful or not is by performing gel electrophoresis. Okay, when you observe like this, you know, okay, the expected size memang kita agak nak dapat dalam 122 base pair. So this is the, we could say that this is the correct band, the expected band that we hope to get. Tapi dia masih lagi dalam bentuk kualitatif, just based on your naked eye. Right? You don't know, you cannot confirm whether, you still have doubt whether okay betul ke tak ni aku punya gene of interest ni. Kan kita tak tahu. Actually you can perform Sanger sequencing. Send your sample for Sanger sequencing, then you can get the sequence of the nucleotide and blast using basic local alignment search tool as the free web server for you to blast the results, so that later you can identify what is the bacteria if you are trying to get the bacteria species. So there are many types of PCR. Macam yang saya tunjuk tadi tu, the step tu is actually for common PCR, traditional PCR. So RT-PCR pula is used to amplify the gene of interest from RNA. RNA is reverse transcribed to complementary cDNA using reverse transcriptase enzyme. So the one that you know when we perform COVID-19 test using RT-PCR tu sebenarnya adalah RT-qPCR. It's a combination of 2 and 3. Dia perlu di reverse transcribe sebab SARS-CoV-2 ni dia tak ada DNA. dia cuma ada genetic material in a form of RNA. So it need to be converted to cDNA first. And then we use qPCR machine or real-time PCR machine to quantitatively determine whether this is a SARS-CoV-2 or bukan. Yeah? Sebab di Malaysia ni, di hospital di Malaysia ni, kita guna RT-qPCR ni sebab kita tak nak dah buat gel electrophoresis. Sebab tu makan masa kan. Kita nak result yang cepat. So by using RT-qPCR, you can actually straight away know whether this sample has SARS-CoV-2 or not by just looking at the graph. Kalau RT-qPCR ni, dia ada screen pula. Dekat mesin dia tu, dia ada screen yang dia akan tunjukkan nanti. Dia punya apa kita panggil graph lah, graph. So dia akan tunjuk pattern. If there is a positive signal that show the presence of the virus, then it will show. Dia akan naik lah tinggi macam tu. So just based on that indicator on the screen, we can know whether the sample has SARS-CoV-2 or not. Macam tu ya. Yang kita ada di hospital tu adalah RT-qPCR. Okay, so multiplex PCR amplifies multiple target in a single PCR experiment. Tengok clue dia je. Multi maksudnya lebihlah daripada satu. Yang saya tunjukkan tadi, kita hanya amplify only one gene of interest. But for multiplex PCR, we can put in several primers, different primers to target for different gene of interest. Okay. So I think this is my last slide. Semua masih boleh bertahan lagi ke?
Boleh.
Okay, so ini saya tunjuk contoh supaya you all dapat gambaran. Saya bagi contoh yang ni sebab ini adalah salah satu projek student FYP saya sebelum ni. Okay, di mana dia collect sampel daripada leaf button ya. So dia gunakan cotton swab untuk collect sampel, and then from there she calculate the colony forming unit on the plate. So this involve microscopic and macroscopic observation on the plate. Yeah. But later we want to know what are actually the bacteria that reside on the leaf button. So we need to do molecular based technique in order to identify the bacteria. So what she did was she need to extract the DNA. Lepas dia extract DNA, dia perform PCR. PCR tu dia pakai primer 16SR DNA. So bila dia amplify PCR, dia dapat size of the expected product in about 1.5 kilo base pair. So bila ranjal kita dapatlah macam ni. Dekat tepi sini adalah dia punya marker, molecular marker. Dia ada ladder, banyak ladder. Setiap senggat tu dia bagi tahu berapa size dia. So daripada situ kita dapat tahu berapa size kita punya sampel ni. Kalau macam ni kan, ini sampel daripada koloni 1, koloni 2, koloni 3 dan seterusnya. Walaupun saiz dia adalah 1500 base pair, tapi setiap sampel ni kemungkinan adalah daripada spesies yang berbeza. So once we've obtained the PCR product, we send the sample for sequencing to company lah yang buat ni kan. Okay, now this is the result of the sequencing reaction. After we obtain the results, we need to analyze using what I've mentioned earlier by using bioinformatic web server. So we analyze the sequence and then we can identify the bacteria up to species level. Macam tu ya. So sebenarnya kalau ikutkan the molecular technique is we cannot rely only to one technique. We need to support this with other molecular technique as well. As of this study, we use two approaches, PCR as well as Sanger sequencing. Okay, so I end the slide now. Do you want to ask anything before we dismiss? Sorry ya, saya ingatkan saya boleh habis cepat. Rupanya dalam 2 jam juga kita habis. Before that, I want to share the Padlet. Let us answer this question first. What do you know about the term hybridization? Okay. Ada yang jawab kat sini combination of two processes, mixture or combination. Ini tak tepat ya. Lepas daripada dengar lecture saya tadi, mungkin ada yang nak bagi tahu jawapan. Siapa yang dengar dengan tekun ni? Jom kita nak habis cepat. Siapa boleh jawab? Hybridization ni apa tadi?
Ligation.
Tak dengar lah.
Ligation.
Ligation. Okay. Lagi? In which single-stranded DNA or RNA molecule anneal to complementary DNA. Yes. Sebenarnya dia kita boleh gunakan term anneal bind. Dia melekat ya, melekat. Ligate pun boleh, tapi ligate tu biasanya kita gunakan additional resource macam ligase, enzim ligase. Enzim ligase tu dia berfungsi untuk melekatkan sesuatu. Contohnya macam plasmid dengan PCR product. Kalau kita nak buat cloning kan, we use enzyme ligase to ligate these two together to form circular recombinant plasmid. But in this case, hybridization is more on complementary binding between the single-stranded DNA tadi tu. Yang single-stranded dengan single-stranded tadi tu dia berjumpa dan dia bind together, anneal. So that is the meaning of hybridization. Yeah. So dia hybridize dengan apa? Single-stranded DNA tadi tu kita punya template, dia hybridize dengan kita punya probe ataupun kita punya primer. Dia punya term tu tadi kan. Dan biasanya probe atau primer ni dia dah tag dengan something. Contoh macam radioactive tag, fluorescent tag. So that kita akan guna tag ni sebagai indikator lah untuk kita tengok nanti dia akan bagi warna ke kan. Macam tu dia akan tunjukkan warna hitam macam radioactive tadi on the X-ray film, kita akan nampak seketul warna hitam. Itu. Macam itulah sebab kita nak bagi nampak dia punya visual tu, so that we can detect based on our naked eye. Dia macam mana dia punya pelekatan tu berjaya ke tak berjaya, macam itulah Okay, apa lagi? From where DNA or RNA can be fished out. Ini sebenarnya daripada mana-mana living things. Macam tadi saya ada sebut semen, blood, dekat pokok, dekat akar boleh, dekat daun boleh, buah, semua boleh. Air, dekat dalam air kita boleh jumpa di surface of the leaf button pun ada DNA of the living things lah. Contoh kalau macam bakteria kita boleh dapat lah. Tapi kalau di dalam manusia apa-apa pun boleh. Urine, di rambut, di najis kita pun boleh, semua boleh. Okay. As long as it is derived from the living things. Okay, to perform PCR, one must have the following items. Ini saya dah tunjuk dekat dalam slide, ada jawapan dia. Application of PCR. Ini ada ke saya sentuh tadi? To identify kan, to identify bacteria as well as to identify the resistant genes, for example, the pathogenic bacteria. We want to also check for the antibiotic resistant pattern. Semua ni ada dalam PCR. Macam kita nak check for COVID-19 tu kan, we want to check for the presence of the virus. So kita boleh gunakan PCR. Yang ini different ni saya dah jawab juga tadi So I hope that conclude this lecture. And if you have anything that you don't understand, you can just come and see me, or you can just WhatsApp me so that I know and I can answer your question. So if there is no other question, we can close this session with Tasbih Kafarah and Surah Wal-Asr. Okay, thank you very much for listening. Assalamualaikum.
Thank you, Dr.
Thank you, Dr.
Okay, you are welcome.